Hi guys, we're going to talk about the cloning cycle. So this is kind of a multi-day cycle that anybody who's worked with bacteria is very familiar with, and I just want to kind of go over the basics of it. And since it's a cycle, it's kind of hard to pick a starting point, but I like to just start with DNA. So we'll assume that you have two tubes filled with DNA. So tube 1 and tube 2, or we'll call them tube A and tube B. And these have a bunch of plasmid DNA inside of them. So sorry, my poor art skills really should be better at this. But we have a bunch of DNA in each one of these. You know, this tube A has part A and tube B has part B. And this has already been QC'd, sequence verified, and mapped, and this is where we're going to start, from QC to DNA. And the type of a cloning cycle that we're going to be doing is the assembly. So let's just draw a little diagram of our parts. So here's our plasmids. We have A, B. These are our part A and part B. We have our eco, our bagel, our BAM, our zo. Eco, bagel, BAM, zo. And we're going to start by digesting. So if we're doing an assembly, we're going to do, what are we going to do? BAMZO, BagelZo. We're going to then run it on a gel. So should be doing arrows here. So we start off in tubes. Tubes go into digestion. So digest. After the digest, we run it on a gel. So here is a gel. So run on agarose gel. And I have tutorials on how to do all of these techniques. So just look them up if there's any questions on that. So then here's our wells. Always have a ladder. Always have a ladder. Run. We're gonna like get two bands. You're gonna have to figure out what parts you need. So we're actually gonna scroll up a little bit. We're gonna say that part A, which is right there, B. We're gonna say that part A is the large. That's what we want, and part B is also the large. So then you run in an agarose gel. And then you're going to cut out those fragments. So you're going to cut them out. So you're going to take a scalpel, and then you're very carefully going to come in and cut around and get as much of the band as you can and as little of the agarose as possible. And then those little gel fragments, they go into a tube. So you get an epi tube, looks kind of like that, with a gel fragment in the bottom. So then you're going to do a gel purification. So gel purification. And that, I have a tutorial for that as well. That's going to be, you're just adding the ADB. So ADB melts the gel, and then you run it on a column. So we have our tube, we add in our ADB buffer, then our fragment dissolves and we get a liquid. It's kind of yellow. At this point, if you're doing a small fragment cleanup, which we're not for this example, you'd also be wanting to add a little bit of isopropanol in there. So we're adding ADB. It goes at 55 degrees in order to melt it. So we come back in 10 to 15 minutes and we get this nice big soup of melted agarose and DNA. And then that goes onto a zymo column, which looks something like that. Ooh boy. And we put our liquid through it, we spin it down. So zymo. And this is all part of gel purification. So these are sub-steps. So we zymo, which is going to consist of putting our mixture into the column, spinning it down, washing it with PE buffer, 250 microliters twice. We're going to dry it. We're going to do a spin drive for 90 seconds. And then we're going to elute it. So we put our column into another tube. So let's see, how am I going to draw this one? We have another bigger tube like this, which also has a cap. And then we squirt a little bit of water. So we squirt water after we've washed and dried. And water goes in there, and it collects the DNA, and then it goes through our membrane, and then it seeps into here. Actually, it doesn't seep. It gets forced into there by the centrifuge, so this happens really quickly. And then we have a tube with digested DNA. So now if we were to kind of diagram what we have, and we've done this for fragments A and B, so let's change colors back to light blue again. So we have two tubes, A and B, with purified digested DNA. So we're going to have A with a digested BAM site. So that means we have that little overhang thingy. And then we have that zoocyte, which has also been digested, with those sticky ends, or exposed sticky ends. And then we have an eco and a bagel site. And that's all A. And then B, we have something pretty similar. So here's our part. Here's the rest of our plasmid. We have our exposed sticky ends for bagel, and we have our exposed sticky end for zo, and then we have a BAM site there. So this would be the end of the gel purification. So we dry and then elute in water. I generally just elute in about 15 microliters, but that really depends on what you're doing. And then you go into the ligation. So we'll change colors for that. So next step is ligate. And all we got to do is take equal amounts of this, and I usually just use 4.3 microliters of each one. A little bit of buffer, so that's actually, so 4.3 microliters of DNA, 1 microliter of buffer. And again, there's tutorials on all of these things, so just look it up. And this is ×2 per fragment. And then half a microliter of ligase. You ligate, and looks like I'm out of room, so we'll start at the top again. So you ligate, and then you get a plasmid. Actually, I don't know if I want to do that. So you ligate, and then you have your full-on plasmid. So let's see here, which is going to be A, B. You have that scar sequence in between. Eco, bagel, BAM, zo. So that's what your ligated plasmid looks like. And then you go into the transformation. So next step is transform. And that's basically where you get your DNA. So that's your plasmid going inside of bacteria. And if this is your bacteria's genome, it then gets a plasmid. And so transformation just consists of heat shock, or you can also do a electroporation, but generally we do heat shock in this lab. And then you're going to plate on the antibiotic resistance after you've recovered, unless you're using AMP, and then you end up with colonies. So then, after plating, you get colonies. So you get a plate, a bunch of colonies on it. From here, you pick your